an official orientation on that matter. So, Cláudia Nunes, também, e João Pedro Silva. Muito bem, ninguém tem falta. Muito obrigado por terem vindo. Portanto, como sabem, as intervenções são de 7 minutos, irá haver um aviso aos 5 e depois, a partir dos 7, uma pequena tolerância minha, mas não muita. E a ideia aqui será fazermos as intervenções seguidas, fazendo uma pergunta no final de cada sessão. Portanto, no final de cada sessão faremos uma pergunta, portanto é uma oportunidade única. Quem quiser fazer uma pergunta pode logo fazer uma pergunta imediatamente e de seguida passamos para a apresentação seguinte e deixamos as perguntas para o final. Ou melhor, podemos fazer duas perguntas no final de cada sessão e depois deixamos as restantes para o final. E dito isto, vamos iniciar. O tempo não é muito, portanto, João, Mação, por favor.
A gente chega lá.
Aqui. Está certo. Bem, boa tarde a todos. Eu sou a Leonor, este é o João. Nós viemos apresentar-vos aqui o projeto Metamorfoses, o nosso tem por nome Som do Silêncio. Este projeto Metamorfoses destina-se a alunos do secundário que tenham assim, uma curiosidade especial, quer seja pela escrita, quer seja pela ciência. Mas pronto, para este projeto ter assim, uma metamorfose perfeita, usando aqui um bocado o nome do projeto, procura uma combinação entre esta componente da ciência e da tecnologia, mas também, portanto, pela escrita e a literatura. A sua finalidade é, então, associar esta parte da ciência, literatura e escrita através de uma construção de um protótipo biónico que servirá como pretexto para uma criação de um conto literário. O projeto resulta, então, de uma parceria entre o Plano Nacional de Leitura, a Ciência Viva, a ESER Portugal e o Gabinete de Comunicação de Ciência da Fundação Champalimau. Então, o que é pronto, passando a coisa, o que é que consiste então o nosso projeto? O nosso projeto, primeiro, como tem por nome, no geral, Som do Silêncio, e o conto tem como título o ensaio sobre a surdez, que tem por referência José Saramago, que tem o ensaio sobre a cegueira. Basicamente, iremos construir, então, uma mão biónica, porque no nosso conto acaba por mostrar a história de que o mundo, ou metade da população, ficou surda e comunica apenas por linguagem gestual, e esta mão tem como objetivo, através de movimentos, fazer letras da linguagem gestual portuguesa e transferir para uma aplicação com uma linguagem meio que corrida, escrever as letras com espaço e assim as outras pessoas possam entender exatamente o que é que esta quer dizer.
Então, como a minha colega disse, o objetivo é, de certa forma, ter esta luva conectada a uma aplicação, ou seja, vai ser uma luva que vai ter incorporados vários sensores e cada sensor, um sensor para cada dedo. E quando nós mexemos a mão, os valores vão mudando porque ela está programada em C++ para Arduino. Portanto, vai estar conectada via Bluetooth a um dispositivo que tenha a nossa aplicação criada por raiz, e essa aplicação vai ler os dados recebidos pela luva e depois, consoante os dados recebidos, ele lê qual é que é a letra da linguagem nacional portuguesa, da linguagem gestual portuguesa, que corresponde. Ou seja, o nosso objetivo final é, neste conto, nós temos uma frase final, que é quem és tu, Joana? E essa frase nós vamos ter que fazer juntamente com a nossa luva, e é esse o objetivo final, é mostrar que conseguimos conectar a aplicação com a luva e vai dar tudo certo. É isso, basicamente.
Pronto, eu não sei se há alguma dúvida acerca do projeto, também se tiverem algumas sugestões, porque o projeto ainda está a decorrer, ainda estamos em fase de programar a luva e dar os toques finais, mas pronto, podem-nos sempre contactar pelo e-mail, não sei. É isto.
Muito obrigado aos dois, parabéns pelo vosso projeto. Bom, vamos então dar início ao painel com a professora Helena Soares, da Universidade Nova de Lisboa, com uma apresentação sobre vacinas de RNA mensageiro, Covid-19 e leite materno.
Ok. As apresentações é suposto estar. Não está a ligar ao... Agora já está. Já reconheceu. Obrigada. Good afternoon. My name is Helena Soares, and I'm the head of the Human Immunobiology and Pathogenesis Lab. In my lab, we have two main research axes. On one hand, we want to know how the T-cell response varies throughout the human life stages, from pregnancy to children to the elderly. And on the other side, we want to know how the same T-cell response varies in different disease settings, from immunoprotection to chronic inflammatory diseases and viral infections. Transversal to these two axes is our keen interest in understanding how tissue specificity is achieved, and in the lab, we are assessing how the T-cell response is modulated by the distinct tissues in which it occurs, from the synovium to the liver. The end goal is to identify therapeutic targets and clinical biomarkers. And of course, a perfect example of immune response compartmentalization is the one afforded by the mammary gland. During pregnancy, T and B cells that are in the periphery migrate to the mammary gland, where they seed, and these local T and B cells are responsible for the production of 95% of the antibodies present in the breast milk, with only 5% of milk antibodies coming directly from the blood. And it is thought that these T and B cells that are in the mammary gland are transported to the breast milk together with antibodies that they secrete, and they play a very crucial role in tailoring the immune profile of the breast milk in order to suit the needs of the nursing infant at a time where its immune system is still very immature. So as soon as the COVID-19 vaccines were introduced in Portugal back in December 2020, we started to addressing this question of what type of immune protection would be transferred from the vaccinated lactating women through the breast milk to their infants. And we expanded our cohort in the summer of 2021 when the vaccination program was extended to the general population. We assessed for each participant, we analyzed paired samples of milk and blood at 3 different time points: before vaccination, 10 days after the vaccine first dose, and 10 days after the vaccine second dose. And our first question was, since the COVID-19 vaccines are administered intramuscularly, if they would be able to prime a local immune response in the mammary mucosa. And we could assess that if we look into the milk and look for an antibody type that is called secretory IgA, that is only produced in the mucosa. It's not produced and is not present in the blood. So the presence of secretory IgA shows that this antibody is produced by the breast mucosa. And in fact, we could find secretory IgA that recognize the spike protein, that is the antigen conveyed by the mRNA vaccines, in the breast milk of all the study participants. Our next question was, how functional were these antibodies present in the milk? And as expected, after the 2 doses of vaccine, all the blood samples had neutralizing antibodies. However, if we assess directly in the milk, only one milk sample was neutralizing. What we thought was happening was that the milk contained more diluted antibodies. So what we did next, we purified milk IgA, milk IgG, and we did the neutralization assays. And under these circumstances, 3 milk samples became neutralizing, which suggests, even though the milk contents are weakly neutralizing, that cumulative transfer of IgA might confer neutralizing protection to the infant. And neutralizing antibodies are important because they are the ones that prevent the virus to enter our cells. And as I said in the beginning, in addition of antibodies, T and B cells are also transferred through the breast milk. Unfortunately, we could not detect B cells at all in the breast milk. However, when we looked into the blood, we saw something very interesting. Vaccinated women that were breastfeeding had higher amounts of memory B cells and of antibodies than vaccinated women that were not breastfeeding, suggesting that the hormones responsible for lactation are playing a role in their immune response. We were more lucky in the T cells, so we could not detect T cells in all the samples, but in all the samples that T cells were present in the milk, we could detect the T cells that are reactive to the spike, meaning that they were induced by the vaccine. And this opens a very attractive possibility, since antibodies only confer a very transient protection in the child, but T cells, as they are long-lived, might confer long-lived and long-term protection to the nursing infant. And of course, this has to be shown by longitudinal studies. So as a summary, we showed that mRNA vaccines can induce antibody production in the mucosa, and particularly in the mammary mucosa, that immune protection through breast milk occurs in the form of anti-spike or vaccine-specific secretory IgA, IgG, and T cells, that milk secretory IgA induced by the vaccine is very weakly neutralizing. However, repeated feedings might lead to cumulative transfer of neutralizing secretory IgA to the nursing infant, and that vaccination induces distinct responses in people that are nursing, in women that are nursing versus women that are not. And this work was made under the LS for Future Consortium by Nova Medical School, IGC, and IQQB. We want to thank Maria João Mourin's lab at IGC, and Marco Alenquer, Margarida Archer's lab, and Hugo Tid at IQQB, and especially to Juliana Gonçalves that led this project. And I don't know if it was me or not, but I'm over, so that's good.
Thank you very much. So we'll have 2 questions. So first question. We can leave also other. So is there any question at the moment for this presentation? No, so I think I might just make a general question. So in the end, how do you evaluate this inherited immune response by the babies? Well, what can you say about this?
Okay, if it is indeed transferred to the babies, that's the question. Yes. So we showed that it's in the milk. Most of the studies, as it is understandable, I can answer in Portuguese if that is better for the audience. Do you prefer? Okay. Are done in animals. And in animals, we know that both antibodies and cells are transferred, and the cells go and see the different tissues. There is indirect evidence in humans that the same also occurs, but of course, looking for cells in a baby derived from the mother is a bit more challenging, and then we have the question or the doubt if those cells were transferred by breastfeeding or if they were transferred during gestation, because microchimerism also occurs during gestation. Going probably a bit more to your question, this is feasible, but it's a bit hard, at least the antibody part. And what another group did in the United States following our work and other studies is showing that these antibodies transferred through breast milk have a duration of 3 to 6 months protecting the babies, or the babies that are nursed by mothers that were vaccinated have an advantage of protection compared to babies that were not nursed by the mothers. And they also could detect the antibodies in the babies until 6 months after birth. But the high would be at 3-4 months after birth. I don't know if I answered to your question.
Yes, you have.
Okay.
Thank you very much. We will follow up to Pedro Moraes, Laboratório de Inteligência Artificial Aplicada. Please. Now I'm following up your lead on the English. That's okay. I hope everyone is okay also. But then again, if the presenters want to, of course, present in Portuguese, that's still up to, it's all okay as well.
I can speak in English, it's okay for me. Okay, so, hello everyone. My name is Pedro Moraes, and I'm here from the Applied Artificial Intelligence Laboratory, the 2AI, and today I will talk about the work feasibility and accuracy of an automated three-dimensional echocardiographic analysis of left atrial appendage for transcatheter closure. So I'm not sure if you are aware, but the cardiovascular diseases are the main cause of death worldwide, and currently we are using more and more technology to improve minimal invasive interventions and to improve all the techniques that are used for cardiology. And all of us has a small appendage that is connected to the left atrium. This small appendage is like a bag, but when we have some pathologies like atrial fibrillation, typically our cardiac function is modified, and the blood flow inside of these appendages start being not normal and starting to create some thrombus. And currently it is known that the major source of thrombus in patients with atrial fibrillation is this structure that is termed left atrial appendage. The small bag is not so relevant. It's only relevant in the embryonic stage. After is only really an appendage. And basically, currently it is quite common that it is required to perform the closure of this small structure by putting a device like this white one that was here presented to stop the blood communication between the left atrium and left atrial appendage. However, the LAA is a quite complex structure. It's curvilinear, tubular structure, and it's quite challenging to perform the correct device sizing. There are multiple devices with different dimensions. And most of the clinical practice are based on 2D image and 2D measurements. In our laboratory, and for the first time, we presented a software to automatically perform the segmentation of the LAA in 3D. The software is able to perform in less than 1 minute. It can be used during the intervention to first create the segmentation and the 3D model of the LAA, and second, to perform the device sizing and extract a set of relevant clinical measurements to detect the optimal device. So we construct all this software during one PhD program, and after that we go for the clinical validation. We integrated all this software in a user interface, and we established a collaboration with a hospital in Hong Kong to validate our software. So this study was published this year in 2022, and basically we performed a comparison of the standard practice that is manual against using our software. And our software could be used in two different ways. It could be used what we term like auto mode, where basically the operator looked for the automatic result, or for the interactive strategy, where basically the operator, after seeing our result, could correct from small corrections to confirm if everything is correct or not. So we selected 158 patients that were retrospectively evaluated, and for all these patients we have the information about the intervention result, and we also collected information about what is the optimal implant for this specific intervention. And with the collaboration of a clinical team, we started in the first time to ask them to perform 2D measurements, that is the standard clinical practice. After that, we asked them to perform 3D measurements too, and then it was also used our software, using the different modes. In terms of results, when we look for the different implants, the accuracy to correctly identify the implants, it was proved that the interactive method, that is this green bar here, achieved the higher accuracy. So when they were using our software, they could correctly identify the device in most of the cases. In 90%, when re-evaluating all cases, in 90% of the cases when using our software, it was much more accurate. The standard practice, that is a 2D, it was only 68. That proved that it's quite variable between observers. And in specific centers where skilled observers could perform the measurements, the manual measurements in 3D, the accuracy was around 83%. Moreover, in terms of computational time, we proved that by using our software, it could be faster. So the standard, this is the blue one, is again the automatic result. The interactive result is the green bar, and the yellow is the manual practice. So basically, using our software, it was possible to be faster and be more precise to perform all these measurements. And as a final conclusion, although we have developed like a fully automatic method, we proved that using the interactive approach, basically a collaborative approach, where our automatic measurements shows a first result, and then the physician could perform small corrections based on the clinical knowledge, we can obtain the best result, and it's the best practice to improve the clinical practice. Thank you all for your time, and I would like to present all our partners on this clinical study.
Thank you. So we have 2 questions. So the first one. So if there aren't any questions at this stage, you can always have more questions in the end of all presentations. But again, I'll make a general question. So how receptive, how willing are surgeons to use these tools? How?
It's quite simple. They have this problem. So basically, they are performing the measurements during the intervention using 2D image, because they cannot use 3D image. To process one 3D image, they need approximately 40 minutes. So they need to perform the measurements like in 2 or 3 minutes, so they cannot have the 3D measurements. With our software, they can have these 3D measurements in less than 1 minute. So they can improve the clinical practice, and they are happy using these results. And we are moving now for prospective studies with a clinical partner here in Portugal to prove the added value of this software.
Okay, thank you very much. Then I think, is there any further questions at this stage? Okay, if not, I think we can move on. Thank you again, Pedro Moraes. And next speaker is Ricardo António Esteves de Castro of Universidade de Coimbra.
Ora, muito boa tarde a todos. Gostaria de começar por agradecer à organização por nos dar a oportunidade de apresentar este tema. Portanto, eu vou-vos falar de cocristais farmacêuticos e mais especificamente de nanococristais. Portanto, o que é que são isso de nanococristais? Quando um medicamento, uma droga vai para o mercado, no estado sólido, ela pode se apresentar sob diferentes formas. Portanto, sob a forma amorfa, portanto, sem muita organização, sob a forma de diferentes polimorfos, uns mais estáveis que outros e com diferentes propriedades, sob a forma de solvados e também uma das formas preferidas da indústria sob a forma de sais. E é das formas preferidas da indústria porque resolve muitos problemas de solubilidade. No entanto, esta forma de sal que não é viável para todas as drogas. Portanto, se uma droga não tiver um grupo funcional e ionizável, não podemos transformá-la num sal. E surgem aqui, então, os cocristais. Portanto, é uma superestrutura molecular com interações de hidrogênio e mais fracas. E este tipo de estruturas já está presente no mercado desde os anos 60, embora este mercado os cocristais só foram regulamentados, os cocristais farmacêuticos, a partir de 2014, 2015, tanto pela Federação Americana das Drogas, FDA, como pela Agência Europeia do Medicamento. Portanto, só a partir de 2015 é que surge o primeiro cocristal farmacêutico regulamentado. E teve bastante sucesso, todos eles até têm bastante sucesso, e desde 2015 até agora estes são os medicamentos que estão no mercado. Em termos de patentes, também podemos ver que os cocristais não estão só envolvidos na indústria farmacêutica, existem também outras indústrias em que ele está presente, na indústria de pigmentos, sabores, explosivos, agroquímica e de nutracêuticos. Portanto, onde é que nós nos focamos na nossa investigação? Procuramos drogas cujo principal problema, então, é o da baixa solubilidade, portanto, de acordo com o sistema de bioclassificação farmacêutica, drogas da classe 2. Arranjamos um sistema bem nosso conhecido, já publicado, do naproxeno, anti-inflamatório não esteroide com a nicotinamida, um coformador inócuo. São aqui as estruturas, e portanto o que nós fizemos foi a formação desse cocristal e associar a essa formação a redução do tamanho de partícula para uma escala nanométrica. E com isso, juntar duas vias de melhorar a solubilidade. Portanto, esta síntese é feita por mecanoquímica, em que ambos os componentes são adicionados no estado sólido, por moagem. Depois, em 2009, descobriu-se que se esta mecanoquímica for assistida por uma gota de líquido, há aqui uma catalisação do processo e o processo é muito mais rápido. Para dar um exemplo, passa tipo de 30 minutos de reação para 10 minutos. E o que nós introduzimos aqui no processo, no nosso grupo, foi a adição de surfactante nesta gota de líquido. E este surfactante vai impedir que as moléculas, que os sólidos recém-formados, portanto temos aqui a ver do cocristal, aumentem de tamanho, continuem num tamanho bastante pequeno. Outra possibilidade é também fazer por wet milling, e nós adicionamos também surfactante ao wet milling. Portanto, os pós foram caracterizados pelas formas habituais, infravermelho, calorimetria diferencial de varrimento, raio-x de pó e os tamanhos por microscopia eletrónica e difração laser. Aqui é bastante visível o efeito da nossa moagem com surfactante. Portanto, as fotografias obtidas por microscopia eletrónica com as mesmas ampliações, e vê-se que a moagem assistida por líquido, as partículas são muito maiores. Portanto, nós fizemos também difração laser e verificamos que, de facto, o tamanho era muito menor, na ordem dos 4,3 micrómetros, e verificamos outra coisa, que ao fim de 30 dias, as partículas obtidas por moagem assistida por líquido começavam a aglomerar-se, enquanto que as partículas com o surfactante mantinham-se estáveis. Portanto, o que é uma outra indicação muito boa quanto à sua solubilidade futura. Por wet milling conseguimos uma nanodispersão com partículas muito pequenas, de facto na escala nanométrica, agora sim, e não verificamos diferença entre a moagem com surfactante, nem na estabilidade. Portanto, em jeito de conclusão, conseguimos a síntese de nanococristais com sucesso. Verificamos aqui nos surfactantes que têm diferentes efeitos e por isso utilizamos uma mistura de surfactantes, até pelas propriedades da molécula e do coformador, e portanto havia surfactantes que faziam crescimento numa direção, outros noutra, e portanto tivemos que misturar com diferentes concentrações para controlar o tamanho das partículas. Portanto, eles foram estáveis por 2 meses e foi uma forma realmente rápida e eficaz de obter nanococristais. O trabalho foi publicado muito recentemente, os editores gostaram da publicação e incentivaram-nos a fazermos a capa, e assim o fizemos. Muito obrigado.
Muito obrigado, professor Ricardo Castro. Temos novamente a oportunidade para perguntas. Não havendo perguntas, talvez eu faça novamente uma pergunta geral. Como antigo cristalógrafo, eu sei a dificuldade de fazer cristais e agora esta ideia de fazer uma estrutura ordenada em 3 dimensões com 2 moléculas diferentes. Queria comentar algo sobre essa dificuldade e os protocolos para obter um cocristal.
Para os cristalógrafos é mais difícil um bocadinho, porque são moléculas muito maiores, não é? Mas nós fazemos, quando é assim, fazemos diagramas ternários para tentar perceber qual é a melhor zona de concentração para conseguir cristais bonitos. Para os cristalógrafos, porque eles não gostam de pós, não é? Embora ultimamente também exista a microscopia eletrónica, a difração de eletrões, e que permite, e estamos a tentar por essa via também resolver a questão, quando não conseguimos cristais bonitos, tentar resolver estruturas a partir do pó por microscopia eletrónica.
Está certo, muito obrigado, professor Ricardo Castro. Não havendo mais perguntas, passamos então, let's move on to Maciej Kozilo, of Institute de Biofísica e Engenharia Biomédica of University of Lisbon. Thank you. I hope I have your...
Okay. Hi, so my name is Maciej. So I am a postdoctoral researcher at IBEP, which is part of the science faculty of the University of Lisbon. And in a truly academic manner, the title of my talk is probably longer than my talk itself, so apologies for that. But I'm going to talk a little bit about what kind of research we do at IBEP, and I do specifically at the Biomedical Neuroscience Lab. And you might have heard of oxytocin, which is a neuropeptide that occurs naturally in our brains. But it's also easy to synthesize in the lab, and we can put it in a spray bottle, in a nasal spray bottle, and we can give it to people to inhale. And there was a lot of research using this compound, using oxytocin. And oxytocin has been implicated as a key player, as a key modulator of social behavior. So there was a lot of hype, actually, and there was a lot of hope that if we have something that we can give in a spray bottle to people, maybe it will help with social impairments in autism, schizophrenia, in other disorders as well. So there was a lot of hope, but when people went and actually tested if it works, if oxytocin helps people, the results were very mixed or downright negative. For example, there was a recent study showing that social functioning was not improved in children with autism. And we think that one of the reasons for that is actually our limited understanding how oxytocin affects cognition through modulating changes in brain activity. So there's surprisingly a lot of questions that are left unanswered still. Our approach is this. So in the brain, when the neurons communicate with each other, they generate an electrical current that can be picked up with EEG, with electroencephalography, like you can see here. This is my colleague Gonzalo, and I'm applying the EEG to him. So we can record the brain activity, which you can see in the middle, and then that signal can be reconstructed into different frequencies. So in other words, neurons talk to each other at different speeds, kind of, and from the signal we can kind of deconstruct and look at different frequencies like gamma, beta, alpha, theta, and delta. And these frequencies, they've been a subject of research for a while, and we have shown that they are linked to some cognitive processes like attention. So for example, if I'm starting to focus on some task, like speaking in front of you, the activity in the alpha range of oscillations goes down. So there's a link with cognition and link with behavior as well. And surprisingly, the characterization of the intranasal oxytocin effect on those neural oscillations has not been very much investigated. There has been just one paper. There's been two papers. One appeared in 2019, and one appeared just last year, and it's by us. So there's really not much research in there. So this is what we do. One of the things we are doing is using the so-called resting state paradigm. So this is a very simple paradigm. So all people have to do is just to sit still, and we record the EEG, and we deconstruct the signal. And of course, we do it after people inhale the oxytocin, and other people inhale the placebo. This is important because it's a really easy paradigm to do in a clinical setting, so you don't have to have any complicated study. People just sit down, we record a bit of activity, can be even 5 minutes, and from that you already have a very rich EEG signal that you can then correlate with some cognitive functions. So we did that, and just to give you an example of some results, we found that after inhaling intranasal oxytocin, we saw a decrease in the oscillations in the theta range, an increase in the theta oscillations. You can see the blue line is after inhaling oxytocin. The beginning of the experiment is at the far left. On the far right, it's the recording after about 1 hour and 40 minutes. So we're actually doing a longer recording just to see how long the effects of oxytocin, those oscillations, last. And we find also some effects in the alpha range, that the amplitudes are decreased throughout the experiment. And you find some other results, but the bottom line is that oxytocin does seem to change the oscillations. And why is this important? This is important because near oscillations, some of the features of oscillations are very stable throughout lifetime. And we have also, as I mentioned before, we also have a lot of research that has linked the oscillations to specific functions. So maybe we can use that to kind of like to characterize people that are more likely to respond to oxytocin. So for example, if you have people with autism that have decreased, they have some impairments in some oscillatory range, and you know that oxytocin is increasing that oscillatory range, for example, then you know it's a more specific target for intervention. So maybe this will help in future to generate better studies and more specific targets for the intervention, instead of trying to just use oxytocin in expensive studies and seeing whether it works or not, which we've seen it's not always the best strategy. So this is it. As I said, the talk is shorter than my title. I just wanted to acknowledge Marie Zelenina, who I co-authored the other study on oxytocin and EEG. And she's also co-authoring the study of the results that I've shown you. This is my contact details if you want to contact me, and if you'd like to hear more about oxytocin in a different context in terms of sexual objectification and neuroscience of sexual objectification. My colleague Carlota Cogoni's talk is tomorrow at 3:30, so you're very welcome to attend as well. So, thank you.
Thank you, Maciej. So, we have opportunity for 2 questions. So if again we don't have questions, you can ask your questions at the end, and again I'll make a very, very general question, which is you are experimenting with human subjects.
Yes.
So I will ask you to talk a little bit about ethics aspects of these kind of experiments, and how the informed consent and what is involved, very briefly.
Okay, very briefly, it's kind of easy because EEG is non-invasive, so we just put electrodes and you can record the activity. And the oxytocin itself also doesn't produce effects that are visible, so you can inhale placebo, you can inhale oxytocin. You wouldn't be able to tell a difference. The differences are very are gentle at those doses, and there have been lots of studies showing that it's completely safe, there's no side effects. And what we do, you apply to the ethics community. That study that we did was actually at ISCTE, so the ethical board has to review the study and agree to it. And we got the green light because even though it involves human subjects, it's very safe procedures, basically. So yeah, it's like this. So you ask the ethical board if it's okay, And with this sort of methods, it's non-invasive, so they usually give you a green light. So.
And did you experiment with one of your co-workers, if I understood correctly?
Yes, when we were like testing the equipment, we just put Gonzalo on the chair so that we can test everything. Yeah, and also I think all of us were doing EEG like ourselves. Yeah, so we tested ourselves and we're fine. So.
Okay, thank God.
Yeah.
Thank you very much. So, thank you, Maciej.
Thank you.
Then next speaker is Cláudia Nunes, from Laboratório Associado para a Química Verde of Tecnologias e Processos Limpos of Universidade Nova de Lisboa.
Ok, muito boa tarde. Eu gostaria então de apresentar o meu trabalho, que é intitulado Combinando Nanoestratégias para a Erradicação de Biofilmes Bacterianos. Como todos nós sabemos, a ciência em geral, e em particular a medicina, evoluiu imenso ao longo dos últimos anos, e em particular no campo dos implantes e dos dispositivos médicos. E, efetivamente, eles são fantásticos, porque conseguem melhorar a mobilidade das pessoas e, em certos casos, até a esperança de vida e impedir que a pessoa morra de uma forma muito precoce. Mas, efetivamente, estes implantes têm um grande, pequeno problema associado. É que existe uma pequeníssimos micro-organismos que facilmente se ligam e colonizam estes implantes. Particularmente no caso das bactérias, elas são capazes de aderir e depois crescer numa comunidade que se protege muito bem, que forma à sua volta uma matriz que impede inclusivamente a penetração dos antibióticos, o que limita muito a sua eficácia, e por outro lado as bactérias que estão no interior têm também um metabolismo mais lento, o que também impede que os antibióticos tenham eficácia. E tudo isto leva também a mecanismos de resistência bacteriana. Isto é tão difícil de tratar que, efetivamente, todos os tratamentos envolvem uma cirurgia, quer seja para a raspagem destes biofilmes, quer seja mesmo para a remoção dos próprios implantes. E, como é lógico, tudo isto tem custos imensos para os sistemas de saúde, já para não falar para os custos a nível pessoal dos pacientes. E, portanto, é isto e é aqui que nós nos fixamos e é isto que nós queremos mudar. Então, basicamente, nós propomos várias estratégias de tratamento destes biofilmes baseados em nanopartículas. E a primeira estratégia, nós queremos encapsular a N-acetilcisteína, ali chamado de NAC, que basicamente é um agente mucolítico, que a ideia é romper a matriz deste biofilme para que depois o antibiótico livre, a moxifloxacina, possa ter um maior efeito. Numa segunda estratégia, em 2 passos, nós vamos usar este mesmo nanossistema, que foi desenvolvido na primeira estratégia, mas com um antibiótico encapsulado. E aqui neste segundo passo, vamos juntar isto a um agente antifúngico, que é a caspofungina, que tem a ação comprovada e sinérgica com a moxifloxacina. Tudo isto, estas partículas são funcionalizadas com aminoácidos, com capacidade de direcionamento para o biofilme e, ao mesmo tempo, com capacidade de disrupção. Todos os ensaios que eu vou apresentar, nós trabalhamos sempre com as partículas com diferentes graus de complexidade, para, no fundo, vermos qual é que era o efeito de cada componente, mas como nós podemos ver através destes gráficos, todas as partículas tiveram um efeito quer na redução da biomassa, quer na viabilidade das bactérias do biofilme. E esse efeito é maior de todos para a formulação mais complexa. Nestas imagens de SEM também podemos ver que as partículas relativamente ao controlo levaram uma dispersão das bactérias, com comunidades muito mais pequenas de bactérias e um biofilme muito menos espesso. Depois, quando fizemos ensaios em combinação com o antibiótico livre, aquilo que nós verificamos é que, efetivamente, todas as formulações tiveram um efeito ainda maior, e este é maior para a formulação mais complexa de todos. E, apesar disto já ser um bom indicativo e um bom caminho, aquilo que nós quisemos testar a seguir foi, efetivamente, será que isto melhora com o encapsulamento do antibiótico, da moxifloxacina? E aqui neste gráfico nós podemos ver, efetivamente, a combinação das duas formulações. E aquilo que vemos é que a diferença mais significativa é, efetivamente, quando temos as duas formulações combinadas. Nestas imagens confocal, em que podemos ver aqui um biofilme como controle, isto é um ensaio de morte-vida, estes pequeninos pontos vermelhos que vocês veem aqui correspondem a bactérias mortas, e aquilo que nós verificamos é que, efetivamente, estas formulações, para além de dispersarem as bactérias, já causam também alguma morte. Passamos, então, para o segundo passo da segunda estratégia, com as partículas de caspa ao fungino, e aqui, mais uma vez, nós vemos que o efeito combinatório com as partículas de antibiótico tem um efeito acrescido. Então, estes resultados impulsionaram-nos para fazer ensaios in vivo e começamos por ensaio de biodistribuição, mas que já tínhamos o catéter infectado, e aqui nestas imagens vocês podem ver estas manchas vermelhas em que efetivamente correspondem às partículas marcadas, mas que se localizam efetivamente na zona do catéter infectado. E podemos verificar também que esta colocalização é tanto maior quando temos as partículas funcionalizadas. Fizemos, depois, a seguir, também um ensaio aqui não de biodistribuição, mas de eficácia, e foi um ensaio que durou 8 dias, e aquilo que verificamos ao fim de 8 dias é que, efetivamente, apesar de termos uma diferença significativa para as formulações mais completas, ainda não tivemos uma erradicação do biofilme. No entanto, quando fomos estudar a existência de bactérias nos órgãos vitais, aquilo que verificamos é que nestes 3 órgãos, para a combinação das duas formulações, não existia qualquer bactéria que se tinha espalhado. Portanto, nós não conseguimos erradicar o biofilme, mas conseguimos evitar que ele se dispersasse por outras zonas do corpo. E isto pode efetivamente ser muito importante, porque uma das questões é que quando se rompe estes biofilmes, podemos levar a infecções generalizadas com consequências muito más, como todos nós sabemos. Portanto, com este trabalho, nós efetivamente conseguimos melhorar a atividade dos antibióticos, dirigi-los ao biofilme, romper a sua matriz e até poderemos evitar a disseminação. E então, as nanopartículas lipídicas mostraram-se ser uma ferramenta para conseguir lutar contra estes biofilmes. Não chegamos a um resultado ainda de completa erradicação, mas estamos no bom caminho. Eu gostaria este trabalho está inserido num projeto financiado pela FCT, e gostaria de agradecer a todas estas pessoas e instituições envolvidas e obrigada pela vossa atenção.
Muito obrigado. Temos novamente espaço para 2 perguntas.
Vocês têm ideia dos mecanismos moleculares que fazem com que este biofilme, o envelope lipídico facilite a destruição do biofilme no fundo?
Sim, pronto, lá está, eu não tive muito tempo para falar da formulação em si, não é? Mas basicamente aqui o que leva à disrupção do biofilme é a funcionalização que nós temos à superfície da partícula, ou pelo menos numa primeira fase. E estes aminoácidos estavam descritos já na literatura por efetivamente se conseguirem ligar ao peptidoglicano e conseguir destabilizá-lo. E parcialmente é por aí que se dá essa disrupção. Portanto, inicialmente por causa desta ação, e depois quando as partículas lipídicas entram e libertam a N-acetilcisteína, como tem este efeito mucolítico, vai ajudar a romper ainda mais esta estrutura.
Muito obrigado. Mais alguma questão? Bom, não havendo, agradecemos à professora Cláudia Nunes, mais uma vez, a sua apresentação. E passamos ao próximo orador, João Pedro Silva, da Unidade de Ciências Biomoleculares Aplicadas, da Faculdade de Ciência e Tecnologia da Universidade Nova de Lisboa.
Porto.
Como?
No Porto.
Do Porto. Peço desculpa. A verdade é que há muitas FCTs no país.
Está. Okay, so good afternoon. Thank you for your kind introduction, and also like to thank UCBO and the organization of this meeting for the opportunity to present the work we've been developing at our lab at the Faculty of Pharmacy of University of Porto to elucidate the molecular basis of mood and cognitive adverse events. Sorry. So adverse events can often occur during and can occur very often during at any stage of a new pharmaceuticals R&D pipeline. However, some of them, like the mood and cognitive adverse events, may only be directly attributable to a drug during the late clinical trial stages or even during post-marketing surveillance. This is mostly due to the poor predictability of some serious central nervous system-related adverse events like depression or schizophrenia at the preclinical phase. This poses major safety concerns, as adverse events may significantly contribute to increased morbidity and mortality among patients and clinical trial subjects. It also increases a lot the costs of drug development, mostly due to the clinical trial failures. So, considering that up to today, there are several drug classes, including antiepileptics, muscarinic antagonists, or fluoroquinolones, that have been reported to induce this type of adverse events, our main challenge is to improve the prediction of mood and cognitive adverse events at early stages of drug R&D. For this purpose, we have developed an original methodology comprising a unique workflow as a first attempt to provide a new mechanistic interpretation of neurotoxic effects involved in mood and cognition. So briefly, the concept of our methodology contrasts with the more traditional approaches like systematic reviews or even meta-analysis of clinical trials, as we mined pharmacovigilance database, the FDA adverse event reporting system, FHIRS, for pharmaceuticals that demonstrate a high occurrence of a defined set of MCAs. Then we ranked these drugs according to the number of reports containing such adverse events for mood and for cognitive adverse events based on statistical analysis. And then drug gene interaction databases and tools like the GI database, the BioVista Visit, or OFX, were used to identify interacting genes and candidate pathway elements for each drug. This allowed us to identify 232 genes associated with the FHIRS ranked drugs, which after expert curation based on the literature, we were able to select a total of 120 candidate pathway elements directly associated with MCAs, namely 21 for mood, 62 for cognition, and 37 for both mood and cognitive adverse events. We then performed functional enrichment and pathway crosstalk analysis to elucidate the molecular mechanisms underlying these drug-induced changes. For example, for the functional enrichment analysis, signal pathways associated with each of these 120 CPEs were firstly identified using these two databases, Reactome and WikiPathway. And then pathways were automatically ranked according to their just p-value. Afterwards, genes encoding for the identified CPEs were mapped to known functional information in humans using gProfiler. And we were able to visualize the similarities and differences between these reactome pathways associated with mood and cognition, cognitive adverse events, in the form of these interconnected networks using a software, freely available software called Cytoscape. And we were able to observe a high degree of overlap between pathways leading to this type of adverse events, as noted by the high number of nodes simultaneously colored with green for mood and blue for cognition. We also were able to identify two main clusters. The first one connecting the modulation of NMDA receptors, MAPK family signaling cascades and synaptic neurotransmission, here, up there. And the second one connecting interleukin signaling with intrinsic apoptotic pathway and protein simulation. We also identified pathways related only to mood adverse events, namely the glutamate mediated and trafficking of AMPA receptors that seem to contribute to modulate neurotransmission and activation of NMDA receptors in this kind of adverse events. And another note linking the transcriptional regulation of MACP2, membrane-bound neurotrophic receptor tyrosine kinase 2, with MAPK family signaling. We also identified cognition-related pathways, pathways that were only related to cognition. This seemed to be a regulated upstream of those leading to mood adverse events, like the DNA repair associated and cell cycle regulation pathways. Also the estrogen receptor mediated signaling, and also the synaptic activation, the postsynaptic activation of nicotinic, the nicotine acetylcholine receptors. Finally, we also queried two databases, innate database and import, to identify which of these 120 identified genes were associated with an immune system response. And we observed that about 66% of these genes were indeed associated with immune modulation, suggesting that this may be an important mechanism to the onset of this type of adverse events. Of course, we are aware that our approach has some limitations. For example, it is highly possible that CPEs, namely receptors with different binding sites, may be site-specific regulator. And we also did not take into account the allosteric versus orthostatic binding. It is also possible that a substantial number of potential binding sites for various drugs have not yet been identified. And of course, we did not take into account the influence of external factors, for example, leading to epigenetic modifications. So to conclude, by combining the power of statistical analysis of pharmacovigilance data with the refinement of expert literature curation, we were able to propose a set of pathways and CPEs with a causal relationship to the onset of this type of adverse events. We evidenced the key role played by the immune modulation in mood and cognitive adverse events. We anticipate that this type of adverse event, needs to be defined at an early stage of pathway network. And finally, of course, all this data has to be experimentally validated, which is what we are currently doing. So to just finalize, I'd like to thank our partners involved in this project and to the funding obtained for it. And thank you for your attention.
Thank you very much. So we have opportunity for 2 questions.
In your data mining, did you disaggregate the data by sex, by gender? I ask this for multiple reasons. One, 8 out of 10 drugs that are removed from the market, they are removed because of adverse effects on women, right? And then on the pathways that you identified immune system, okay? And there's a bias in the immune response, and women have a higher inflammatory response than men, and estrogen, so kind of it popped out. And then we could go on about other the social aspects of that and how complaints by women are perceived in the clinic versus by men, but kind of biological only. Did you segregate the data by sex?
No.
You should.
Yes, that's a very interesting remark, and it would be interesting to do so, of course. At this stage, we had a lot of data to mine, and we had to select different stringency criteria. As we were not aware of exactly what we were looking for, at this stage we did not want to sieve that kind of information this early. So we wanted to have all the bulk information before going after these specific differences. We only discarded, not only, but for example, I remember we discarded data based on the age, specifically for infants and for the elderly, because the data is very confusing in these pharmacovigilance databases. It's not easy to directly attribute the effect of a drug to this kind of adverse event, the effect of the drug to this kind of a diverse event in these age ranges. But that's a very good suggestion. Thank you for it.
Outra pergunta? Temos outra pergunta?
How would you use this in practice to predict the adverse effects of a drug that someone is going to take? Can you do it?
That's our main challenge right now. So this was the first step, has to have some insights where to look at. We are currently doing several experiments at different stages with different partners working on it to assess if any of these targets or candidate pathway elements that we identified could indeed be used, maybe not individually, but together in a toolbox to help predict this kind of adverse events. I don't have any data to give you right now because we are, as you may know, with the pandemic, everything got a little bit delayed. But I hope that by the end of this year we'll have very nice results and interesting insights regarding how this identification of these pathways could lead to a better an earlier identification of these adverse events.
Thank you.
So, thank you, João Pedro Silva. And now let's move on to Diana Lousa from Instituto de Tecnologia Química e Biológica, António Xavier, da Universidade Nova de Lisboa.
Can you tell me where the pointer is? Oh, it's not here. Oh, okay. Sorry. Okay, so I'm going to present the work that I've been performing on SARS-CoV-2, namely the development of biopharmaceuticals in ITQB Nova, which is here in Oeiras. So I think you all know, I don't need to remind you, that COVID-19 is having a devastating effect in our lives. And this pandemic is caused by SARS-CoV-2, which is a coronavirus that is wrapped by a lipid membrane and contains RNA that it uses to replicate, and a very important protein known as the spike protein that is important for the virus to enter the host cell. And this protein, it binds to the receptors in our cells, the ACE2 receptor, which enables the virus to insert the RNA into the host cell and then replicate. Okay, here I'm showing you the X-ray structure of the spike protein bound to ACE2, and ACE2 actually binds to a particular region known as the receptor binding domain, the RBD. And we, in the scope of a task force that was launched in ITQ Innova, namely in the Most Micro Research Unit, we are both studying the spike protein using fundamental research methods and also using applied research to develop biopharmaceuticals against this protein. So the idea, in a very simple way, the idea of these tailor-made biopharmaceuticals is to block the spike protein. So basically they are proteins that will bind to the RBD and block the spike protein, preventing it from binding to the ACE2. Okay, so this is the basic idea. And for this, we are using a combination of computational design, so computational methods, and experimental validation. In our lab, we do the computational design. Basically, the idea is to use the structural knowledge that we have on ACE2 bound to the RBD, and then we select small proteins with stable and compatible structure, in this case helical. And the idea is to graft the motif, the ACE2 binding motif, in these stable scaffolds. And then we have an extra step where we optimize the binding of ACE2 to the receptor binding domain, basically by doing mutations and analyzing the binding energy in a high throughput fashion. We have to do thousands and thousands of cycles. Also, we try to optimize the structure of these proteins. And so we scanned a very large library of proteins to choose the best ones. And in the end, we designed 5 RBD interacting chimeric proteins. Chimeric because they have the scaffold and the binding motif of ACE2. And we ended up with these 5 proteins, which are our first candidates. And computationally, we can already predict their fold. I'm not going into the details, but this type of funnel pattern indicates that they should have a stable fold. Also, we performed MD, molecular dynamics simulations, just to see if the binding would be stable. And they show that it is at least stable enough for our starting point. And then these results were validated in the lab. So the proteins were expressed in E. coli, they were purified, and then they were analyzed. They were shown to have, most of them, to have an helical structure by CD. And then they were analyzed by surface plasmon resonance to measure their binding affinity for the RBD. And at least one of them, the first one, had a fairly good binding affinity towards the RBD. And we are now improving these proteins, and then the idea is to get better and better affinity, then measure their ability to neutralize viral infection in cell infection assays. So in conclusion, I think this shows that the computational design of proteins is a promising strategy to develop biopharmaceuticals, not just against SARS-CoV-2, but against different viruses and even bacteria. And these studies contribute to the development of these strategies, which are still a bit in their infancy. We are now following more sophisticated strategies involving not just known scaffolds, but de novo protein design. And we also aim, obviously, to target new variants and even new viruses. And so, finally, I want to thank the people involved in the work, especially Professor Claudio Soares and our lab, the lab where I work, and the people involved, Carlos, Mariana, Susana, Carolina, and also in particular João Vicente, who also with some of them validated our results experimentally, and all our collaborators, and also Most Micro and the LS for Future associated laboratory, who provided the framework where we did all this, and also FCT.
Obrigado, Helena. So we have time for questions now. You can go on to the first question. So if there aren't any questions at the moment, you can ask them afterwards. I think I will make a question, just a naive question. Do you need that these peptides, these drug candidates, have more affinity to the ACE2 protein than the actually receptor binding domain in the spike protein, or it could be about the same or even less?
Okay, so actually it's the opposite. They have affinity for the spike protein.
Yes.
They are kind of competing with these two.
Yes.
Okay, for now they have less, the ones that we have designed, but Baker's lab has designed inhibitors that have higher, significantly higher affinity in the picomolar KD range compared with nanomolar for ACE2. So it's possible to go, because you can optimize them. Of course, for that you need directed evolution and things that we still don't have access to, and money to. No, it's not just computational. You need to optimize things in the lab also.
Okay, so ideally they will have more affinity.
Yes. To be able to really compete and completely block the interaction with.
So you cannot just copy nature. You have to go.
You
have to
should
be better than nature. Yes. So.
Did you did any study regarding the toxicity of these proteins in humans or in cell lines?
We haven't, because they were not good enough for us to spend time and money analyzing the toxicity. But in our protocol, there's a step to evaluate their toxicity first in cell lines, then in animal models. The things that have been coming out from other labs indicate they are not very toxic.
So, if there are any more questions about Diana's presentation, if not, I think we can see if there are any questions about any of the other presentations or general comments about this session. And then again, if not, it's almost 5 o'clock, so I will close the section. Thank you all for coming, and thank you all the presenters for for showing their work, for sharing their work with us. Thank you very much, and we'll see you tomorrow. Thank you.